Hello, and welcome to another educational session with BioIVT. I am Becky Campbell, a technical advisor for ADME Talk Services here at BioIVT. As you can see from my title, To See or Not to See, we will be discussing CYP2C induction today. We will touch on services and product highlights. So what does the CYP induction study do? It assesses the potential of drug candidates to induce the expression of drug metabolizing enzymes and therefore predicts the in vivo DDI potential, or drug-drug interaction potential. This is important because, as you might know, CYP enzymes are the most important class of drug metabolizing enzymes, and it is estimated that over 50% of all drugs are metabolized by this class of enzymes. So if the activity of a CYP enzyme changes, it can have a drastic effect on the metabolism of these drugs. In other words, CYP induction increases the risk for drug-drug interactions. If a CYP enzyme is induced and it increases its activity, that can lead to an increased metabolism of co-administered drugs. That in turn can reduce their therapeutic effect or increase. This simple figure illustrates that principle. On the left, we have the situation under normal or standardized conditions. Drug 1 is taken by a patient. It is metabolized, and in this example, metabolized by CYP3A4. The resulting exposure, clearance, and efficacy is as predicted and intended. Now, if a patient takes drug 1 and drug 2, the situation can change. In this case, drug 2 is a CYP3A4 inducer, which will lead to an increase of CYP3A4 enzyme and normally to an increase in activity as well. As a result, drug 1 will be metabolized faster, and that means that there's a shorter exposure of drug 1 in the body, and therefore efficacy is likely to be reduced. So it's obvious that understanding a drug candidate's induction potential is critical and supports the evaluation of the risk for drug-drug interactions. Regulatory agencies understand the increased risk with CYP induction. Therefore, evaluating CYP induction for CYP1A2, 2B6, and 3A4 is required by the ICH M12 guidance. In addition, if CYP3A4 is shown to be an inducer, it is recommended that CYP2C8, 2C9, and 2C19 induction are also assessed. I want to mention two examples where CYP induction has played an important part in drug-drug interactions. The first example is with oral contraceptives. Estrogens and progesterones, the two active ingredients in oral contraceptives, are primarily metabolized by CYP3A4. A patient needs to be cautious if she also has to take the antibiotic rifampin to treat gram-positive or mycobacterial infections, for instance, like tuberculosis. This is because rifampin is a strong CYP3A4 inducer, so the hormones in the oral contraceptives are metabolized faster and will lower the efficacy, which for obvious reasons might lead to unwanted results, such as an unexpected pregnancy. Another example is St. John's wort, which is a natural supplement used for different indications, such as mild depression. The substance in St. John's wort, hyperforin, is a strong CYP3A4 inducer. As a result, St. John's wort is known to cause drug-drug interactions. One of these is with cyclosporin, which is an immunosuppressant and often given to organ transplant patients. As in the previous example, if St. John's wort and cyclosporin are retaken at the same time, it causes cyclosporin to be metabolized too quickly. This results in a weaker therapeutic effect, and in other words, weaker immunosuppression and possible transplant rejection. I have mentioned that CYP induction is assessed using mRNA levels, and that the ICH M12 requires evaluation of CYP1A2, 2B6, and 3A4. This is because inhibition of CYP activity can mask induction if only CYP activity is evaluated in induction studies. The regulatory guidance requires that if CYP3A4 is shown to be an inducer, that the CYP2Cs, so CYP2C8, 2C9, and 2C19, are also evaluated. With that said, the induction potential is typically lower for the 2Cs than for CYP3A4, and we often recommend using enzyme activity to assess the induction of the CYP2Cs to obtain more reliable results with the 2C positive control. This is especially true for CYP2C19, where activity is actually required by the ICH M12 because of an industry-wide known issue with CYP2C19 mRNA and the positive control response. Here I have gathered historical data for CYP2C induction from several induction studies to demonstrate the differences in response by the positive control between mRNA and activity results. Take note of the CYP2C8 and 2C19 results. We don't see much of a difference in CYP2C9 mRNA versus activity. But as you can see for CYP2C19, the average fold for rifampin was 1.85 for mRNA versus 14-fold for CYP2C19 activity. Additionally, rifampin failed to reach twofold in 71.1% of assays when evaluating CYP2C19 mRNA versus only 1.1% of activity assays. We see a similar trend with rifampin failing to reach sixfold in 100% of assays when evaluating CYP2C19 mRNA versus only 25.7% of activity assays. A similar trend is true for CYP2C8 mRNA versus activity, but not to the extreme observed for CYP2C19. For CYP2C8, the average fold change for rifampin was 4.25-fold for mRNA versus 6.96-fold for CYP2C8 activity. Additionally, rifampin failed to reach twofold in 22.1% of assays when evaluating CYP2C8 mRNA versus only 1.9% of activity assays. Again, we see a similar trend with rifampin failing to reach sixfold in 83.8% of assays when evaluating CYP2C8 mRNA versus 52.4% of CYP2C8 activity assays. This is important to note because when the prototypical inducer positive control reaches sixfold or greater, the interpretation of in vitro induction by a test article is much clearer when interpreting the potential for drug-drug interaction risk by in vitro induction results. BioIBT has several lots of cryopreserved platable inducible human hepatocytes available. Several lots now include CYP2C induction as part of the extensive characterization to help you choose a lot for your particular program needs. As you can see in the red box, induction of CYP2C mRNA by rifampin is highlighted. BioIBT is the leading global provider of ADME products, including human hepatocytes. All of our products are extensively characterized, and our protocol to evaluate human platable hepatocytes now includes the characterization of lots for CYP2C enzymes. This includes CYP2C8, 2C9, and the ever-troublesome CYP2C19. Visit us online at bioibt.com to learn more about our extensive catalog of biospecimens for use in research programs. A search of human cryoplatable hepatocytes will take you to a page with more information on these products. There you will see the Browse Live Inventory button that will display lots with available inventory. Click the Document button for listed lots to see characterization data, including CYP2C19. You can also select and order products, or we can help you with your induction study with the ADME tox services that we provide. Thank you very much for your attention, and I look forward to next time.